It's a great day for Symposium. I'm Geraldine Bliss, and I want to welcome you to the first Phelan-McDermid Syndrome Drug Development Symposium. We are very excited to be in beautiful and chilly Cambridge for CureSHANK's inaugural symposium. So I wanted to start with a few housekeeping notes. Restrooms are located right across the hall, and then there's another set on the other side if you need to find the restroom. There's a coat closet in this hallway right over here. You're welcome to drop your coats or bags over there. If you requested a parking pass, it is in one of two places. It's either stuck to the back of your badge, or if you requested two of them, they would be inside of the badge holder. And if you need a parking pass, and you didn't request one in advance, I think we might have a couple extras, so come find me later. I hope that you've received your amazing CureSHANK swag. I see most of you haven't picked it up yet, but on that table we have some special CureSHANK swag in honor of our inaugural event here. So please be sure to grab one of those before you leave. Speakers, we have a little change. If you're giving a presentation with slides today, I sent you some instructions that now have to be changed, unfortunately. We would prefer for you to give us your presentation on a USB, and if you don't have a USB handy, Talia can help you with that. Okay. So, my son, Charles, he's now 25 years old. He suffers from Lennox-Gastaut syndrome, which began at the age of 8. My promise to Charles to help him get better was my reason for co-founding CureSHANK. We have a singular purpose, which is to accelerate the development of treatments for Phelan-McDermid syndrome. So we're very happy you're here today. When I began my research advocacy journey about 16 years ago, most of the scientists who were interested in SHANK3 were basic scientists, and they were interested in understanding sort of the basic mechanisms of learning and memory, not necessarily for cures for some rare disease. Today, we're at an inflection point where there's a robust set of translational tools for PMS drug development. There's a fantastic PMS natural history study. We have an infrastructure for conducting clinical trials in PMS. We have all of the right things in place. We even have several clinical trials that have already paved the way. But much work still remains. This morning, we want to start by sharing all the amazing work that has already been done, translational and clinical research. And then this afternoon, we will turn our focus to the remaining challenges and chart the future of drug development for Phelan-McDermid syndrome. So before we get started with the scientific program, I want to bring your attention to a few resources. On the tables, you'll see a QR code, and if you scan that QR code, it'll take you to a landing spot where you can find links to today's agenda, the program guide, as well as several resources that CureSHANK has. You can also, if you don't have your phone or want to do it on your computer, you can, let's see, do we have the I have the clicker. Okay, here we go. Okay, you can type in this URL, cureshank.org/PMSDDS 2024, and that will take you to that landing page. You'll find some valuable resources there. We held an externally led patient-focused drug development meeting in front of the FDA about 2 years ago, and you can find the Voice of the Patient report there. You'll also find our latest project, which is an interactive conceptual model. So a couple of years ago, we commissioned a conceptual model of Phelan-McDermid syndrome with a company called Adelphi Values. And we recently took that conceptual model and turned it into an interactive model where we overlaid on comments from families from the Voice of the Patient report. So it kind of gives you a little bit more context for what those concepts really mean and what the impact is on patient and family quality of life. Right now, it only works on desktop browsers, so if you're playing around with it right now on your phone, you won't really be able to access all the features, but you can pull it up on your computer, and then you can kind of play around with it. You'll also see we have some social media resources here. Hopefully, you will be on social media today. You can tag CureSHANK, and our hashtag is PMSDDS 2024. So now I want to take a moment to introduce you to the CureSHANK team. CureSHANK is a parent-led organization. We have a board of 4 moms, and our members are Talia and Marie Silva. Talia is our de facto director of communications as well as our vice president. Paulina Rychenkova, where's Paulina? There's Paulina over here. Dr. Rychenkova is one of the co-founders, and she is our secretary. And we have a 4th board member, Abby Levincy, who's just like the heart of CureSHANK. She is our treasurer and our de facto chief operating officer. And she's running her first half marathon tomorrow in Los Angeles, so she couldn't be here in person. I threatened to break her legs so that she'd have to be here, but it didn't work out. So that is our sort of our executive team and our board. We are very fortunate now to have 2 staff working for CureSHANK. The first is Anna Mingorance, who's our new Chief of Translational Science. And we brought Anna on because there's so much happening now in Phelan-McDermid syndrome, and we really want to enable efforts by companies, and Anna's here to help us with that. So we're grateful for her. And Trish Davidson, there's Trish over there in the corner. Trish is the program director of our CureSHANK Biomarkers and Outcome Measures Consortium, and this is a consortium for companies, for anyone who's interested in developing treatments for Phelan-McDermid syndrome. And the idea is to work together on projects of mutual interest, things that can enable greater success in drug development in PMS. And then I also want to introduce you to our advisors. We have several advisors that guide our science. The first I want to recognize is Brett Abrams. Where's Brett? Please stand up so we can see you. Okay, great. And Federico Bolognani, right here. And Eric Hoffman, where's Eric? Oh, over here. Walt Katoniuk. Is Walt here yet? I think he's showing up a little bit later. Morgan Shang. Here's Morgan. And then our board also, our advisors also include Tom Frazier, Shafali Jeste, and Jamil Beg. And Jamil's wife is having a baby this week, so he's not here. So I also want to extend a special welcome to the parents who are here today. If you're a parent of a child with PMS, would you please stand up? Great, y'all. Okay, wonderful. Thank you for being here. I just want to point out that all the parents have a special badge. It's yellow, and we did that so that if you're a scientist or a member of industry, you can seek out the parents and chat with them. There's so much to be learned from the parents about the lived experiences of PMS. So thank y'all for being here today. And that brings us to the first talks of our scientific program. We're going to start with some talks from 2 parents, moms. And I want to begin by introducing you to Tiffany Aaron and Joanna Kent. Tiffany and Joanna, they're moms, and they're going to share their family stories. So we're going to get started with Tiffany first.
Hello, everyone. I'm here to speak to you today on behalf of my 4-year-old son, Brooks, who was diagnosed with Phelan-McDermid syndrome at 11 months old. Like most moms, I had genetic testing when I was pregnant, and everything was normal. Life couldn't be any better. So here he was, my beautiful, healthy 9-pound baby boy. As milestones approached, I had an excuse for why he wasn't holding his head up or wasn't reaching for toys or rolling over. Then there was the doctor visit when I heard the words, further genetic testing. And then I got the call. I remember the feeling I had, and to this day, I get the same gut-wrenching feeling when I hear the words Phelan-McDermid syndrome. The more I read, the more I couldn't believe what I was reading. Moderate to severe intellectual disability, seizures possibly, aggression. No, not my baby. These things don't happen to us. Well, they did. And everything I hoped wouldn't be in our future soon became our reality. I gave up my sales career I had worked so hard to excel at to be able to now be there for my son because I was going to do everything. I was going to make everything better. We were going to do every therapy. Maybe this therapy will help with speech or functional independence, or maybe, just maybe, if we did enough therapy, he could catch up. The thing is, this disease impacts everything in the individual affected, not just fine motor, gross motor, neurological, feeding. It affects the ability to stay alive. I cannot tell you how many times we've had to call 911, a number that I never had to use before. The seizures are terrifying. The rushing of doctors towards your son's hospital bed, using the bag, and then saying we have to intubate. Looking at your 3-year-old on a ventilator is a heart-wrenching feeling no one should ever have to experience. Continually hearing from doctors that there are no answers because this isn't a typical seizure. And the dreaded words that I also learned from Google that play over in my head, there is no cure for PMS, haunts me daily. I worry about what will happen to my intellectually disabled child if he outlives me. This room is filled with such capable, educated people, and I feel honored to be able to tell you just a glimpse of our story. I also want you to think about what it'll be like if you were in my son's shoes. He has things to say as well. He wants everyone to know that he loves them, but he can't tell them. He wants to know why he can no longer say mama when he is hurt like he used to be able to. And I'm sure he wants to know why he wakes up to these grown-ups sticking him and poking him in a hospital bed that he can't get out of. I'm sure he also wants to know why he can't play like the other kids at the park for fear of falling, or why, when he tries to, they usually just walk away. I can't think about these things for too long because they hurt more than anything you can imagine. So I want to ask this room to commit to developing treatments for Brooks and others with Phelan-McDermid syndrome who so desperately need it. If anyone could do this, I know this group can. Thank you.
And this is Joanna Kent.
I was not expecting to have to follow that. Hello, my name is Joanna, and I want to thank you all for being here to listen to our stories, share information, inspire hope, and break medical boundaries that impact more than just our children. From the moment we got our diagnosis, I shared it with the world, my family, friends, our community, complete strangers. I've shared information, struggles, triumphs, and today's not about the triumphs. Today is about the reality that being a supermom, or being told how strong I am, is not inspiring. It is depressing. It's only a reminder that this is not the person I want to be. This is not what I wanted for my son. But the true story is about when I lost him. Within a few months of getting our diagnosis, he had his first regression of skills. I knew exactly what was happening as I slowly watched my son disappear. I'm always thoughtful of explaining it because it wasn't a regression. It was a complete and utter loss of devastation. My once adorable, sweet, gentle, slow, but excited to copy your movements boy was gone. And with him died my dreams for him, myself, and my family. I became a shell of myself as he bit me, and scars appeared along my body. He pulled my hair so much that I had to cut it all off because I had balding patches. He kicked, elbowed, punched me, and I silently took it. I took care of this person who didn't know I was their mother, and I became numb and lost accepting my fate. I am terrified of the future. My life, my husband's, our daughters, our lives have been altered by genetics in a way that isn't fair and it isn't right, and it's confusing. My daughter doesn't have a brother. She has someone who steals all the time and attention of her mother. My husband doesn't have a son. He has a burden. I gave up my career, my hopes, my dreams, and my life is all about someone who will never know and will continue to hurt me physically and mentally while never having any idea they're doing it. When your child is the 1% of the 1%, You see the world differently. Most people in my position, they care for the child 24 hours a day. And any minute they aren't, they're hoping for a miracle. I'm hoping that things don't get worse. My plea to everyone in this room today is that small changes are so huge for people who depend on others to feed them, bathe them, toilet them, help them walk, track everything they eat and drink, who can never be left alone because they're a danger to themselves physically, and they're unable to communicate if anyone actually harms them. The list goes on. For a moment, imagine that families didn't have to give up careers and ambitions or become dependent on the government for programs and help because their child could open their own doors. At this moment, it seems like a dream, but I believe in science, and with your help, you don't just help those with Phelan-McDermid syndrome, but their siblings, grandparents, parents, and their communities. You help us all. So thank you again for your time, and I look forward to the day that we have effective treatments with your help.
I'd like to introduce you to MUSTAFA SAHIN. He is the director of the Rosamond Stone Zander Translational Neuroscience Center at Boston Children's Hospital. Many of you probably already know Dr. SAHIN because of his pioneering work in tuberous sclerosis. His lab also has an interest in SHANK3, and very importantly, he's also the he's the consortium director of the Developmental Synaptopathies Consortium, which you're about to hear a lot about. So MUSTAFA, and I guess I'm going to let you come up and introduce your speakers.
Thank you. Well, as Geraldine mentioned, I've been working with a number of different rare genetic syndrome families and patients over the last 25 years, and I just want to say this room, the motivation, the passion, the resources that are in this room are really exceptional, and I hope we can all learn and make progress today. We're going to take on potentially the biggest challenge in my mind in improving the lives of individuals with Phelan-McDermid syndrome and reducing the suffering affecting these families, with the big question being, how can we do clinical trials in a way that will show effectiveness? How can we get FDA, EMA, other agencies to approve treatments that will have an impact that will be covered by insurance? That's sort of the end goal we want, and how do we get there from a clinical perspective? So we have 3 presenters in the first session, followed by a panel discussion. First is Dr. TESS Levy, who's going to tell us about the genetics and epidemiology of this condition. Tess?
Hi everyone, I'm Tess. I work at the Seaver Autism Center in New York, and I'm excited to be here. I will be briefly talking about the clinical features of PMS, but we'll be spending most of my time talking about the genetic causes of Phelan-McDermid syndrome, what we know about epidemiology, and then what we can predict for the future in terms of epidemiology based on what we've learned from our research group. So starting briefly, we all know that PMS is a neurodevelopmental disorder. Developmental delay and intellectual disability is probably the only feature that we see across every individual with PMS, but the severity of that can differ. Other very prevalent features are things like problem behaviors, medical features like hypertonia, sleep problems, and GI problems. A lot of the current research today, at least at our group, is focused and also modeled after top caregiver concerns. So things like seizures and epilepsy, which have been associated with premature mortality in some kids with PMS, and then skill loss, regression, and adolescent age psych changes, which Alex will be talking about later today. Moving on to genetics, we know that the cause of PMS is heterozygous loss or haploinsufficiency of SHANK3. This is either caused through a 22q13 deletion of SHANK3 or a sequence variant or mutation in the gene. Looking first at the deletions, we can see that the size varies a lot. So some kids have deletions that include just SHANK3, and other kids have deletions that include SHANK3 and also more than 100 other genes. You can see that also by looking at the size of the deletions. So some kids have deletions under 10 kb and others have over 8,000. Deletions currently are usually diagnosed by microarray, But more recently, they've also been reporting back on exome and genome sequencing, and historically, and also still in some places, karyotypes are used to detect these deletions, but those can really only find the bigger deletions and miss some of the small ones. There are two special types of deletions I thought were worth mentioning. The first is a ring chromosome. So for these kids, in addition to the Q arm deletion, that includes SHANK3, the top of their short arm is also deleted, and it makes the chromosome form into a ring. And because of the instability of the ring chromosome and another gene on the chromosome, these kids can have more medical risks than some of the other kids with PMS. It's hard to know the real prevalence of ring chromosomes because not everyone who has a microarray to diagnose the deletion will then have a karyotype to look for the ring chromosome, but at least in our cohort, it's about 10 to 20% of kids with deletions who also have a ring chromosome. The other type I thought was worth mentioning are unbalanced translocations. So some parents have a balanced translocation where they have the right amount of genetic information, it's just not stored or organized in the way it should be. And when they have a child, there's a chance of the child inheriting an unbalanced version of that. The reason it's worth mentioning is because we can see recurrence in the same family, so more than one child with PMS, or at different generations of the family, many individuals with PMS. Again, hard to know the true prevalence because not every parent receives follow-up testing after their child is diagnosed, but it seems to be lower than ring chromosomes, so maybe around 3 to 5%. Looking at the mutations, so these are picked up with any sort of gene sequencing, so panel sequencing, exome or genome. The very large majority are protein truncating variants or predicted loss of function variants, the most common being frame shift, nonsense, and then there's also a handful of splice site variants. As a special consideration, I put missense variants just to say briefly that we should be careful interpreting these. For the most part, they're usually classified as uncertain or benign, but recently we've seen a few cases that have been accidentally classified differently. So just a note to say we should focus on the protein truncating ones. This, I hope, just illustrates the variability that we see across the genotypes in PMS. So the first plot shows you the mutations mapped onto SHANK3. The y-axis here is the count of individuals who have a mutation at that site, and then the x-axis is the amino acid position. So this just shows that there's mutations throughout the gene, and there's one very recurrent mutation that we see, which is the frame shift mutation you see as the tallest bar in this plot. On the other side, you see all of the deletions mapped onto chromosome 22. So each individual bar here is one individual's deletion. The very top blue bar is the largest deletion in our group, and then the very bottom pink bar is the smallest deletion. The blue and pink here is just class 1 and class 2 deletions, which is a way that we separate these for some of our analyses. And then at the bottom, you see all of the genes in the area. So again, you can just see the variability in terms of size and gene content of our kids' deletions. So most people, I think, when they hear that there is a wide clinical spectrum and also variability in the genotypes, they wonder if we can relate the two. So our group and many other groups that look at PMS have spent some time looking at genotype-phenotype relationships. Again, first to see if we can understand some of the variability we see clinically, but also to help parents have a better personalized understanding of what they could expect for their children. I think, at least in my opinion, one of the biggest lessons that we've learned from all these studies is that the loss of SHANK3 really is the main culprit here for all of these features. So whether kids have small deletions, big deletions, or mutations, they all have kind of, in some senses, very consistent pictures. On the other hand, there are some very clear genotype-phenotype relationships that we have seen. I think the clearest are some of the medical features that we really only see in kids with bigger deletions. Kidney problems, I think, are the most clear, where in our group at least about 25% or a quarter of the kids with bigger deletions have some sort of renal abnormality. and then people with smaller deletions and mutations never do. So that just points to genes other than SHANK3 that are responsible for these specific features. The other consistent finding that we've seen across our group and other groups is that kids with larger deletions tend to have more severe developmental delays and intellectual disability. So they tend to achieve less milestones, things like speaking in words or phrases, and if they do achieve them, it's often much later. On the other side of this, we also see more reports of regression in kids who have smaller deletions and mutations, but we're still trying to tease apart if they have some sort of increased risk based on their genotype, or it's just a factor of they achieved more milestones to begin with, and therefore we're able to measure loss in these kids. We're also seeing more reports of adolescent age psych features in kids with smaller deletions and mutations, but again, we're still trying to understand if that's something to do with genetics or if we're just better able to appreciate these symptoms in kids who have relatively more skills. Things like autism, problem behaviors, and many other features of PMS, we see consistent rates across all genotypes, which again just points to SHANK3 being the most important gene in this region. For epidemiology, so this I was asked to talk a little bit about. I think especially with genetic disorders, it is difficult because we really don't know everyone who's not diagnosed with this condition. So I have a bit of a roundabout way before getting there. First is a very brief history of PMS. So from 1985, we have the first case reported. Over the next 15 years, researchers worked to narrow down the candidate region and candidate gene, which they found as SHANK3. And then we started seeing published case series of SHANK3 mutations, and then finally publications that included both deletions and mutations under the umbrella term as Phelan-McDermid syndrome. And I put gene therapy 2024 because we are all excited about that. So this, I hope, just illustrates, I think first that we really haven't known about PMS for all that long, and second that we have known about deletions for longer than we've known about mutations. The second piece, which I think is an important part of this story, is the standard of care for genetic testing for kids with developmental delay. Starting in 1975, that was a karyotype, but honestly, I don't know how many people actually received those. In 2010, that was changed to be a chromosomal microarray, and then by 2019, the standard of care was recommended to be an exome. I put recommended because it's been very slow for insurances and the government to actually approve exome for most kids who have developmental delay. So for this piece, it's just important also to see that while we've known about deletions for longer, we've also been able to test for deletions and diagnose deletions longer than we have mutations. And really that genetic testing is the main barrier to understanding how many kids really have PMS. And then a second point to really just hammer this home, this is a simplified table from a project that Audrey and I have been working on with the CDC. These are Medicaid claims from 2008 to 2016 for kids 7 to 17. And they look to see if these kids received any genetic testing at all in their history. So we should be seeing 100% for all of these groups, but for kids with autism, only 17% of them received genetic testing. For kids with intellectual disability, it was lower at 13%, and then kids with autism and intellectual disability, only a quarter of them ever received genetic testing. So again, I think these numbers are, I think, a lot worse than we were expecting to see. But again, I think the lack of accessibility of genetic testing provides like an illusion that all these genetic disorders are very rare when in fact they aren't. That said, I can talk a little bit about what we know about epidemiology. So there's a few places I thought that we could find relatively unbiased numbers from. The first is genetic testing labs who are able to provide how many kids they see or they test with developmental delays and how many of those have PMS. Ambry Genetics, which is a lab that I prefer these days, provided numbers to me on April 1st. So for kids who got a microarray at their center or at their lab, 1 in 1,000 of them were diagnosed with PMS. and then 5 out of 10,000 of kids who were tested with some sort of sequencing for developmental delay were diagnosed with PMS with a SHANK3 mutation. If we add these together, which isn't perfect because there's some overlap there, but to be conservative, if we add them together, we can say that 15 in 10,000 kids who get genetic testing for developmental delay have PMS. And the next part is a guess, so I'm not trying to really prove these numbers, but if we know that about 1 in 50 kids have intellectual disability in the US, and 15 in 10,000 of those have PMS, then we can estimate that perhaps 1 in 30,000 individuals have PMS. There could be about 10,000 in the US and perhaps 250,000 worldwide. So these are definitely not the most accurate numbers. I think the point I'm just trying to make is whether this is 1 in 30,000 or 40,000 or 50,000, it's certainly not 1 in a million or something that's very, very rare. Some place that we can look for some actual proof, though, the Phelan-McDermid syndrome registry provided numbers that were updated also, I believe, of April of this year. This has national and international enrollment, and they've been doing this since 2011. So far, they have over 3,500 families in the registry, and something that happened in 2023 and consistently so far in 2024 is they have 1 new family registering with the foundation per day. This shows overall the world, so there's no real reason we should be expecting different rates across different countries. In terms of this registry, it's likely, 1, just accessibility of genetic testing, and then also language. I think the registry is available in English, maybe also a few other languages, but not all of them. So the other thing I wanted to show is just some clues that we can take from our research cohort and use them to see what we can expect in the future and what we can learn about epidemiology of PMS. So this group I put together for the next few slides is a combination of all of the kids we've seen at the Seaver Center and then the RDCR Natural History Study phases 1 and 2. So the group comes out to just over 300 people. Males, females is pretty equal, and then in terms of genotypes, we're seeing 2/3 of those with deletions and 1/3 with mutations. And within the mutation group, you can see that 1/4 of them all have the same exact mutation, which is a frame shift at position 1227, which is just very recurrent, and we've so far seen a lot of kids with the same one. The first thing I wanted to look at is the rate of new diagnoses over time. In this graph, the y-axis is the number of individuals diagnosed, and then the x-axis is the year. So looking at this year by year, it's less clear, but if you look at this kind of 5 years at a time, you can see that every year more and more kids are being diagnosed. There was a very big peak in 2014 that I can't really account for. It might be coincidence. There was also a handful of high-impact autism genetics papers that year, which might have contributed to it, or it might just be coincidence. If we look at that same graph and split it by genetics, not surprisingly, for a long time, we're seeing that only people with deletions are diagnosed, which is one, because that's all that was known, and also that's all that we could test for. But over time, we're seeing that more and more mutations are being diagnosed, and by 2018 was the first year that more mutations were diagnosed than deletions. And since that year, it's been consistently that we're seeing more kids with deletions be diagnosed, or more kids with mutations be diagnosed than deletions. So if we compare the last 5 years to the previous 20, we see very different ratios of deletions to mutations. This is just another way to look at that same plot. So this just shows the proportion of kids based on their genotype of who's diagnosed each year. And then the last thing I think that I wanted to show is just the age of diagnosis over time. So these can be important especially when we're thinking about treatments and gene therapies. Getting people diagnosed as early as possible is important. In this graph, the y-axis is the age of diagnosis of the participant, and then the x-axis is the year of birth. The dotted line is how old that child is now, so anything above that is the future, and we won't have information on, so this graph is kind of skewed in that way. But we are seeing at least that more and more individuals are being diagnosed at younger ages over time. Again, if we split this by genetics, we can see that kids with deletions are being diagnosed consistently younger than kids with mutations are, which might just be due to administrative work of getting exome sequencing approved and the time that that test takes, but might also be due to kids with deletions who often present with more severe delays, and they may just present earlier than the kids with mutations, or likely perhaps a combination of both of those. Things I hope you can take away. PMS, I think, is a lot more common than we think it is, and access to genetic testing remains the biggest roadblock, in my opinion. And based on current trends, we know that more individuals every year will be diagnosed with PMS. The hope and what we've seen so far is that individuals will be diagnosed earlier and earlier, and also we'll probably begin to see the ratio of deletions and mutations level off at some point within the next handful of years. And a special note, as of this year, New York Medicaid now covers exome sequencing for developmental delays. So while late, it is definitely a big step in the right direction of seeing more kids diagnosed. Thanks to everyone here, and probably many other people not on this slide, and also especially to our PMS families who bring us lots of joy every day at work.
Oh, it's so nice to be here, and it's so nice to be in person. My gosh. You know, Geraldine has for like 15 years now asked me to do the impossible, and today she's asked me to, what do you do? I can't see it. She's asked me to present like 1,000 things in 20 minutes. So I'm going to do that. So it'll be kind of like rapid fire, but then Mustafa will ask me questions. I have a couple of other comments. It was really nice to hear from the parents. It was really powerful stories. I was walking over here this morning and struck by the fact that it was a beautiful day. And that's kind of unusual for me in Boston, but it's nice. And I was seeing all these MIT students, right? And I was feeling like walking by all these MIT students, maybe I picked up some extra IQ points. So then I could apply those extra IQ points to the mission at hand, which is to develop better treatments in Phelan-McDermid syndrome. And then I heard TESS speak and thought, wow, how lucky this field is to have TESS contributing and sort of starting her career. You know, it's pretty cool. And she got an early PhD on that slide, which is nice also. Tess is working on her PhD, but...
Which one is
okay. All right, I do have to put my glasses on.
Sorry.
I can't see the whole title.
Oh,
Does that say, yeah, open up? Sorry, I'm not a Mac person.
Sorry. I took a double click. I'll do it. It should open. Jeez. Here we go. So it should open.
That's not it. I do a clicker, right? Oh, this.
Yeah.
What else did I want to say?
Sorry.
Let's see. Oh, our GSC team, right? We have this amazing, like, family that we've developed now over the 10 years we've been collecting natural history data. And it's just been, like, a remarkable group to work with. Ah, thank you. I've had experience with a lot of different consortia. This one's truly special. And, yeah, we're especially pleased to be able to welcome other companies in biotech and our new Jaguar friends to the family. All right, here we go. If I do this, can you see my cursor? No, all right. You'll just have to get oriented. So here are my goals. We'll talk about preliminary natural history results, and I'm going to go through it relatively quickly. We'll talk about our goals in terms of trying to become clinical trial ready, which has been really the main impetus behind the natural history studies. And then I'll give you a little bit of an update about what we know and what we don't know in terms of clinical trials and how we're preparing to kind of run better clinical trials. So these are our kind of overall aims, and this has been since, I mean, for 10 years, really. We kind of modify them a little bit, but in general, we've been really focused on just comprehensively characterizing the phenotype, tracking the natural history over time, developing biomarkers. So the first iteration looked a lot at imaging, and then we got a little bit, what's the right word? Less enthusiastic, shifted over to looking more at EEG markers. And then finally, thinking very clinically about developing consensus guidelines based on what we knew, and then sort of how do we apply that to a practical way of helping families. So this is our amazing team. About 5 sites. I think everybody's here for the most part, right? Liz is always busy. Usually, so okay. Liz is here in spirit. What's that? She's always busy and she's always available? Oh, exactly. Right. So, yeah, I'm sure. So, right, so this powerhouse group, and sort of multiple generations now. We can, I think, call Mustafa the grandpa. Yeah, sorry, Mustafa, but I think it's a distinguished role. Yeah, he started the thing, he's sort of leading it. I mean, the story, by the way, was that Geraldine asked me to just put in this RDCRN grant and wanted PMS to be the main driver, which we of course, were not ready for. But Mustafa was ready for it, so thank you, Mustafa. But we've now collected data from over 200 patients across many, many years. I think we have about 50 patients who have 5 time points, and these are successive annual time points. So this is a really rich data set. I think, in fact, it's probably the data set that was most attractive to the gene therapy companies. And so this is really starting now to yield some really valuable information. God, I can't see anything. So this is an example of our sort of phenotyping battery. You can see that we look at a whole variety of different domains: cognition, adaptive behavior, language, motor function, you know, a range of behavioral symptoms, autism symptoms, and so on. And so we're getting a lot of information from these kids across multiple time points. So as Tess mentioned, you know, we now have a pretty good sense of what the clinical phenotype looks like. Certainly, the common theme is neurodevelopmental disorder, intellectual disability, adaptive functioning deficits, communication delays. And then there's a whole host of other features that are somewhat variable. But these kids all will have sort of moderate to severe intellectual deficits, and that's a main driver of the challenges. I think also important to note here, you know, less than half will develop any kind of meaningful phrase speech, and about 25%, it depends a little bit on which data set you look like, but only about 25% will gain bowel and bladder control. And that's obviously a big deal for families. Lots of other clinical features, the most common of which is hypotonia, gastrointestinal symptoms, which are also especially challenging for families, apraxia, you know, we hear a lot about renal abnormalities, epilepsy, so this is one of the most kind of concerning symptom domains for families. Only about 25% of kids have epilepsy. Probably about 40% will have EEG abnormalities. But among that group, there's a smaller subset that has, you know, very severe refractory epilepsy. The majority, though, seem to be well controlled. There's no like specific seizure phenotype, but the majority of kids with seizures are well controlled. We hear a lot about dysmorphic features. There's a huge variety of dysmorphic features. Some kids have many, some kids have few. I don't think we're going to come to a place where we're going to see some, you know, pathognomonic dysmorphic feature. We're never going to come to a place where a disease-modifying therapeutic is going to alter a dysmorphic feature. So I'm not so concerned about that. Most of our kids are super cute. That's what's important. Autism, you know, those rates have been going down and down and down as we're a little bit more refined in our way of thinking about it. About 65%, you know, now we think meet criteria for autism, whatever that really means in this population. Disrupted sleep is you know, profoundly affecting families and kids and super common. And then we talk about regression a lot. You know, the regression numbers, 25%, 30%, 40% in some studies, depending on the domain. I'll talk about that in a little bit more detail. We're learning a lot more now about like psychiatric changes, especially around puberty, and real shifts in the neuropsychiatric phenotype and what that looks like and what some of the risk factors are, including how to treat it, which has been reassuring. So finally, Audrey, Sid, where's Sid? Kristen. This team has been thinking a lot about actually analyzing the data from what we've collected, and we're now at the point where we can start to do that. And so, you know, one measure that's gotten a lot of attention that we think is potentially very valuable is the Vineland Adaptive Behavior Scale. But there's a lot of ways of looking at the Vineland. The standard way, or the standard score, when you look over time within subjects, This is a study that Sid submitted. It's not yet published. Where's Sid? Ah, I Sid. I think this is is this the first time this is being presented? Okay, so look at this. I get to present Sid's stuff for the first time. Yeah. So the idea here is that the standard scores don't change. If anything, actually, you see a decrease in socialization. So standard scores, when you look at them over time within subjects on this measure, are relatively flat. But if you look at the growth scale values, they do change. They kind of go up a little bit. So if you look at receptive and written communication, those GSVs are actually going up a little bit. Not enough to drive the standard score. Expressive is not going up, unfortunately. And this, by the way, this is just natural history, right? We haven't done an intervention specifically. I mean, these kids are all getting interventions, but in general, these are kind of climbing up, which is a good thing. Personal daily living scores and domestic daily living scores also are going up, but not enough to drive the activities of daily living domain, the broader standard score, right? So this is important, and it gives us a sense of what we want to be using in terms of measuring adaptive behavior over time. Audrey, anything to add? Audrey has very strong feelings about this. How'd I do? Okay, good. Oh, two thumbs up from Audrey. Okay, so characterizing regression. This is John Bernstein's group out of Stanford. This is actually before the DSE even really kind of came to be in a formal sense, but he published this small paper looking just at a couple of questions on the ADIR and alerted us to the fact that there are kids who are regressing, picking up the younger regression. So he described 42 cases, 18 of whom had regression. Average age of onset was 6. I think we now know it to be a bit younger. But it was affecting virtually every domain: motor, language, self-help skills, and so on. This small sample didn't see any association with deletion size, didn't see any association with epilepsy or any kind of trigger specifically, and we're sort of learning more about what might be relevant there. I'm waiting for Tacey's paper. No. So that here we looked at cases with neuropsychiatric changes and loss of skills within that group. There seems to be another cohort later on around puberty or young adulthood who develop neuropsychiatric symptoms and then loss of skills in that context. And those kids actually, once you treat them, some of the skills can be regained. And that's an important kind of distinction between the early loss and the later loss. And now here's Tacey. Tacey's here. Yeah. So Tacey started to get really kind of tuned into this and started to interview parents, specifically ascertaining parents who had children who experienced psychiatric problems. And in this case also, she saw the average age around 15, regression across a whole variety of domains. Here, 50% regained skills, likely because they were being treated, which is a sort of a promising note. And this is one of the first times we were really alerted to the prevalence of catatonia and how kind of misunderstood and often undiagnosed this went. So we looked at our database. This is Kristen Farmer. She's working closely with Audrey. Audrey's picture should have been on here, but it's too busy to slide. So this is a huge database taken from the PMS registry. And what you see is that there are regressions across a whole variety of domains: fine motor skills, gross motor skills, self-help skills, and social skills. And if you look under the first blue line, it's somewhere between 20 to, I guess, 34% of these kids are regressing in these domains. That's significant. If, by the way, you look at the entire cohort, I think the number is 44% of kids had regression in at least one domain. That's a lot, and this is a huge database. I mean, it's all parent report. Nobody's evaluating these kids directly, but it's 44%. I'm looking at Audrey. Yeah. Okay. So this is concerning, but it's also capturing mainly the much younger kids. If you look at the ages, the majority are losing skills at around 1 or 2, but it's not uncommon to lose skills after 10 also. And then the number of kids in this particular cohort, anyway, that are regaining skills is closer to about 1/3. So it's a lower number that are regaining skills among those who lose skills younger, right? But the skills are being lost across all domains. And then looking at our DSC database, this is a cohort of 207. gosh, I can't read this. I think you're at almost half have lost skills, and the most common domain to lose skills in this cohort were the language skills. So for those who develop words or phrase speech, about half lost them, and that's a very big deal. Motor skills, much less likely to lose. Very small percentage of the kids lost skills. Self-help skills were lost, closer into the 30% range. And then social skills for those who gained them, also lost.
I do have something to add here.
Please.
Which is that these are really early developmental skills based on the autism literature, and we're now looking much better, starting to look much better at skills for later kids. So that isn't shown here yet.
It takes a village. Thank you, Audrey, appreciate that. And then, a lot of work of trying to predict who might be at risk of losing skills. And we haven't really clarified this yet. I think what we see is, as Tess pointed out, there's a clear relationship between deletion size and gaining language. So you're more likely to have language if you've got smaller deletions and sequence variants, but you're also more likely to lose language. Is it that these kids are more likely to regress, or is it just that you're more likely to see it because if you don't have language, you can't see a regression? So we really haven't quite figured that out yet. I think the answer is probably going to be both, but we do see a clear relationship with genotype here and language regression. So we need better measures, and that'll be part of the theme here. And one of the cool measures that Audrey's developed, see Audrey's picture is on here, is the LOST survey. So we've now started to distribute this to our entire cohort, and we're collecting data, so we'll hopefully have something cool to present at the next meeting. When's that? Next month? June? July? No. And then also in terms of the neuropsychiatric symptoms, we've been spending a lot of time thinking about neuropsychiatric symptoms in the context of our diagnostic and statistical manual for mental disorders, which is really clearly not relevant to these kids with PMS. So I have this kind of fantasy that we'll be able to create our own little kind of cluster of symptoms and then define them differently, specifically for Phelan-McDermid syndrome. So we have this amazing group, the ECHO group, that meets twice a month, that Tacey and I guess I had something to do to start it. And we meet with family, or with doctors who are taking care of families, and we're hearing a lot about these neuropsychiatric symptoms. And we collected a fair amount of data about it. So we have here presented both the ECHO results for the first 50 cases and results from a psychiatric survey that we developed that was based mainly on the diagnostic and statistical manual of the DSM, and captures sort of the common symptom domains that we think are most characteristic of this population. And so here you see, there's a lot of convergence, thankfully, between what we learned from the parent survey that was distributed online and what we learned from the ECHO, which was just based on doctors presenting cases to us. But things like sleep disturbance and anxiety, depression, mood fluctuations. So we're starting to get a much clearer picture of what the neuropsychiatric phenotype looks like. And thankfully, we're getting a much better picture of how to treat these symptoms. And we're seeing some success, luckily. About a third of these kids, even more, 40%, actually presented with catatonia. This is actually a very, very dangerous, potentially dangerous outcome that we need to do much better to address. So all this information kind of came together. We spent years going through it and thinking, what have we learned over the last, I guess it was 8 years at that time. And Sid took the lead on developing these consensus guidelines. We pushed these out about a year ago, actually. And hopefully they have been a contribution. I think there's been a lot of doctors in the community that have learned to be able to reference these. And that's I think, been a big advance. Okay, clinical trial readiness. Everyone's, okay, we're good. How's my timing? I'm pretty much done? Okay. Seriously? Oh, shit. Okay, we'll go quickly. So the first thing we did was we looked at the standard measures that we always use in the autism literature. So the repetitive behavior scale, the SRS, and we discovered that they weren't so great, right? So in the repetitive behavior scale, overall, kids with Phelan-McDermid syndrome are less severely affected. That doesn't mean that it's a relevant tool. It just means that there's going to be some floors. SRS, we learned. I've said this over and over again. We learn as much from our failures as we do from our successes. So the SRS, not a good tool, right? All these kids were basically ceiling didn't distinguish at all between autism and not autism within this population. So many of the questions are very kind of language-based. So we just can eliminate that as a relevant tool for this group. On the other hand, Paige Seiper and Teresa Tavasoli at the time in our lab got very interested in the sensory phenotype. They created a very cool kind of combination observational tool with caregiver interview, a bit like the ADOS, the observational piece anyway, where they score in a relatively standard way sensory reactivity, so hyposensitivity, hypersensitivity, and sensory seeking across all the different domains, tactile, auditory, visual. And we've done a lot of work with this tool. It's definitely relevant across neurodevelopmental disorders, including in Phelan-McDermid syndrome. And we've captured a pretty specific phenotype in Phelan-McDermid syndrome that reflects hyporeactivity. Obviously, that's consistent with the high pain threshold that we see and parent reports. Companies have started to jump in. So Neuron, which had trofinetide and is now thinking about putting one of their IGF-1 analogs into a larger trial in PMS, realized that we need to adapt our instruments to be relevant for Phelan-McDermid syndrome. So they took a look at the CGI. This has been done in Rett. This has been done in Angelman syndrome. And they applied the same kind of methodology to try to anchor the CGI kind of domains into the PMS phenotype. So they're looking at fine motor, gross motor, receptive language, expressive language, social cognition, a whole bunch of different domains, but they're doing it in a very kind of systematic way, and they're going to eventually apply it. I think already have applied it to their trials. There's both there's a CGI severity scale and a CGI improvement scale. And it's very structured, and it gives the clinician who's evaluating the child lots of opportunity to take notes and then kind of pull those notes into what is a global score. And this is just an example of that. I won't go into a ton of detail. But I think this is a terrific tool, and I'm looking forward to being able to work with it more. Visual evoked potentials. So we spent a lot of time at the Siever Center developing visual evoked potentials. These are derived from looking simply at a contrast reversing checkerboard, just 3 electrodes in the occipital cortex, and you extract this really kind of consistent waveform. Everybody in this room would have the same basic waveform, which you see on the left, where the, I guess that's the N75 peak. It reflects excitatory activity and has got this beautiful little amplitude. It's a nice, strong excitatory activity amplitude. And then the P100 reflects that kind of inhibitory response. So, excitatory glutamatergic activity, and then inhibitory GABAergic response. And it's unbelievably consistent in healthy kids. 2 minutes, lovely. I mean, it's not fair. I mean, we'll just eat into the panel a little bit. We're gonna have to eat into the panel a little bit. Sorry.
Well, is that okay?
Yeah, because I mean,
Yeah.
5 minutes, okay. So, right, in Phelan-McDermid syndrome, however, we see a totally different waveform, which is equally consistent, and you essentially see a loss of that excitatory peak. So you'd expect exactly that because, these SHANK3 proteins anchor excitatory synapses, and so you're flattening out this peak. It is over and over and over again consistent. We look at kids that are typically developing. We look at kids with autism who are idiopathic without a known cause. We look at Phelan-McDermid syndrome. We look at the siblings, and over again, the amplitude of that excitatory peak in Phelan-McDermid syndrome is suppressed. Why is that important? Because you can do pre and post in clinical trials. We've done some very small preliminary studies looking at the effect of IGF-1 as an example on one of the frequencies, and we see an increase in frequency in the gamma domain. You asked then, or Brett's going to ask me, I'm sure, is there a connection then between clinical symptoms? We do see improvements in clinical symptoms as measured by the SAHIN. And then, of course, the most important piece is that you can marry your electrophysiological biomarker with your clinical symptoms, because these biomarkers are cool, but they really need to be related in some way to clinical symptoms in order to be clinically meaningful, obviously. And so we do see that the kind of lower the amplitude of that excitatory peak is correlated with a greater sensory hyporeactivity. So this is kind of this nice story. 6 patients but we're definitely chasing this down. Basically, everyone that comes through our center and gets clinical trials has pre and post VEPs that Haley does. April will tell you about her work you're presenting. Okay, good. So April looks at alpha and gamma power. She sees differences between people with Phelan-McDermid syndrome and healthy controls. This is a sort of a striking evidence that there's a clear sort of phase uncoupling. This is just baseline resting state EEG. And then April will talk to you about the idea that Phelan-McDermid syndrome has an overall positive phase bias, whereas typically developing controls have a negative phase bias. Is that correct, April? This is good. All right. So again, EEG markers, biologically objective, hopefully sensitive to change in response to treatment. Okay, clinical trial. I'll go fast. So it started with the first clinical trial ever, which was IGF-1, which, by the way, is TESS 2014. So that might correspond to your increase in diagnosis. This little paper. I wouldn't call it high impact. We did a follow-up study. What? It was a good journal. Oh, it was great. No, great journal. Yeah, great, good point. Joe's the editor of that journal. I'm not sure how much it was cited. We did a replication study. Then we sort of ran out of IGF-1. It's very hard to get IGF-1. It's super, super expensive. The manufacturer doesn't manufacture it appropriately because it's got only one indication. So we backed off to use growth hormone because we know that growth hormone increases the release of IGF-1. So we did a small study, just sort of proof of concept that showed, yes, you can actually increase IGF-1 with growth hormone, and it was associated with small improvements in a very tiny cohort. Concurrently in China, and I had no idea, they were running essentially the identical trial as us, and they showed improvements in the aberrant behavior checklist. And I also didn't know that the aberrant behavior checklist has been translated to Chinese. True story. Companies jumping in, that was exciting. So IGF-1 analogs being looked at by Neuron. So this is it's a synthetic analog of CGP. The idea here is that it reduces inflammation. It sort of promotes the synaptic integrity. It reduces, and it's supposed to also normalize levels of IGF-1. I'm not sure if that's been looked at just yet. But these guys finished their first open label trial. I think it was with 18 patients or so. With some solid preliminary results. Of course, it was open label, but it seems like they have every intent to go into a what would be a phase 3, actually. Yeah, so it's really exciting. People kind of jumping into this space, and I'm personally thrilled about pursuing IGF-1 or IGF-1 analogs. Another company jumping in. Where's Joe? Joe Horgan's here. So this was the first company, AMO. This is Joe. You're Joe Nolan. That's Joe Horgan. Yeah. Two companies, Joe. Yeah, I'll tell you. This is the first company that had the courage to jump into Phelan-McDermid syndrome, specifically with AMO01, which is an ERK inhibitor. They had some preclinical data. We did a small trial. We showed improvement in seizures, which is super exciting, consistent with what we saw preclinically with the mice. And this has actually been submitted for publication by TESS. I have two more.
Two more slides?
I have three more.
Why don't the panelists come to the front so we can, as Alex is finishing, we can set up the panel.
This is, oh, aren't you going to show Kate's video?
No, we're going to skip that because of timing.
Oh, okay. Not everything has been successful, including translational studies. So where's Hala? Hala had a beautiful SHANK-3 rat that showed clear deficits in synaptic plasticity, clear improvements with oxytocin. We did do an oxytocin trial on 18 kids. We did not see significant differences, unfortunately, with oxytocin. There's studies that are going on in Europe now, in France, in Italy. So there's a lot of activity in this space. We are really excited to see people jumping in here. We're really excited to see them using randomized controlled designs. I don't know if you guys heard about this. There's a little company called Jaguar. They're pretty excited to get into this space. We're really, really thrilled to work with them. We've got our site set up. We're hopefully somewhat clinical trial ready. So that's really fantastic. There's a number of different companies that have been talking about gene therapy. The other one that's gone public, and at least the intent, is PYC Therapeutics. They've developed an ASO, so they're going to be increasing expression on the intact allele. Another really kind of exciting approach. This is to kind of anchor us in the caregiver perspective. This was actually sponsored in part by CureSHANK. looking at what is really important to families. So what do we know is important to families? Communication, cognition, preventing regression, sleep. So these are things that we're constantly being reoriented to, just by way of, I guess, reassurance. I don't need to summarize.
Great.
I do need to thank.
Hi, I'm Pam Ventola. I'm an associate professor at the Child Study Center at Yale, and I'm a chief science officer at Cog State. So I see patients clinically with PMS at Yale, and then I work with a lot of different pharmaceutical sponsors that work in PMS and other rare disorders.
Hi, Lata Valori Polysuria. I'm a clinical psychologist at Rush in Chicago. I work with Liz Berry-Kravitz, and we have a PMS clinic and are part of the natural history study.
I'm Audrey Thurm, clinical psychologist at the National Institute of Health, and I direct the Neurodevelopmental and Behavioral Phenotyping Service. I'm involved in natural history study and other genetic conditions as well.
Hi, I'm Sid Srivastava, a child neurologist and neurodevelopmental specialist at Boston Children's, where I direct our PMS clinic there.
So we've thought about some questions to just get the discussion going, and then we're going to open it up to the group later. So maybe we can start with Pam. We're all interested in sort of minimally clinically important differences. Well, how can we measure those differences in rare diseases like PMS? And I was wondering if you can start the discussion about what are some of the methods to consider for that.
You really start with
Yes.
the easy questions, right?
Yes.
So, your MCID, it is always the question, because in rare disease, our sample sizes are going to be small. We're not necessarily looking for statistical significance. And then even if you get statistical significance, what does that mean? That's not necessarily going to promote payers for the drugs, or it's an answer, it's not the only answer, and I would argue it's not the most important answer. The most important answer is what is clinically meaningful. And this is not, there are no consensus guidelines in rare diseases. You can look at other psychiatric indications like Alzheimer's disease and other, we know what these are, but in rare diseases we don't. So we need to tackle it disease by disease and measure by measure. And there's also not one answer to this question. There's not one way to do this. When I work with sponsors and get asked this question, I really lean on the guidance from the regulators, which relates to hearing the perspective from patients and their families, hearing from key opinion leaders and the clinicians that are treating these kids. So what you can do, one, you can do exit interviews. So you have an early phase trial, kids have improved, you say you ask the families on exit, did your child improve? Was the magnitude of that improvement clinically meaningful for you? Then you can map that on to a numerical value in your trial on that endpoint. So they move 2 points. The families are saying this is clinically meaningful. That is your clinically meaningful difference. I've also been working with a sponsor who's entering phase 3, and they're interested in the Vineland as their endpoint for their key secondary in a phase 3. And what we did was we administered the Vineland to 25 families with the kids that had this rare seizure disorder. So it wasn't PMS, but related. And we said, if there was a treatment in a year, how much difference? So here's your Vineland score now. How much would you need to see improvement for it to be clinically meaningful for you? And it was approximately a gross score value of 2 or 3 points, which is absolutely attainable. So we're going to go, we have a meeting this summer with the FDA to say, we're going to use the Vineland. This is our MCID. This is why.
Great. Maybe Latha, I can ask you about using established scales, some of which we know that don't work in PMS, versus developing new scales, and then also adapting scales to disease-specific purposes. what are the benefits and I guess the risks of doing that?
And I'll welcome all the panel members to speak to that. I mean, I think there's, and this speaks to something else that I think we'll probably get to, is our existing scales, especially for clinical trials, are typically based on psychiatric conditions where we're looking at decreases in challenging behaviors or going back to a sort of baseline. In many of our rare diseases, we're looking at increasing or improving skills. So where we really just don't have scales that are measuring learning and cognition. And the standards, when we think about clinical improvement, the standards can't be the same, right? For when we teach a skill, even for us, you know. Atomic Habits is a really popular book, right? So like they talk about how like it takes like in behavior therapies, and that's my background, I'm a behavior therapist, it takes like minimally, typically developing individuals 6 months to learn a new skill, right? And you think about like our skill gains come from, even without any other challenges, really small changes. And our measures in clinical trials and psychiatry and mental health, just not there. So I mean, I think just pulling back from what we have, we just don't have those. So we do have to develop them if we're measuring skill gain.
Others to comment?
I think that with your question of start building new measures from scratch and adapting measures, both of those things unfortunately take a long, long time to get to where the FDA will accept them as validated, which is why the pressure is on adapting measures, because we think that takes less time. But the truth is, even when you adapt it, you then need to provide all the validation. So, you know, if people wonder why we focus so much on the natural history studies, we are at the same time learning about the disease as well as working on these measures. So, I mean, what Alex showed of what doesn't work is so critical to what we're doing. And then just trying to be very systematic about how, if and how we're adapting, trying to work with the metrics we have first, because that's the easiest thing to do, is just figure out which metrics are best before we work on actually changing anything. Because again, anything you change, you're either breaking standardization of a standardized measure, or you're developing something new, and then you have to work on validating it. So just understanding and patience with that piece of it, because the FDA will not approve a drug if the measure isn't shown to be validated in this group for this way.
Sid, maybe I can ask you about mapping the natural history data that Alex showed that you and others are analyzing, and how to map that onto clinical trial readiness, and how that data could be used as a historical control, if you will, for some intervention.
Absolutely. I think it's, I think one point just to really emphasize that I think Pam had made earlier is what exactly, how to define meaningful change. So in the data that Alex showed, where we looked at the natural history of these Vineland standard scores, but also growth scale value scores, even if there is a positive change, even if there's an improvement of, say, 2, I'm just making this up, but 2 growth scale value points over 1 year of time, what does that actually mean for the patient? Is that what is the functional significance of that? So I think there needs to be a little bit more work into taking some of these standardized assessments, of which we have a lot of data for, and going back and trying to say, well, what, at the end of the day, how does this translate to the families, to the patient, and to functioning? So I think there needs to be a little bit more of that. But at the same time, I think it's really terrific. We have this kind of robust, broad swath of assessments, and now we have to kind of systematically look at each of these assessments, whether it's the Vineland, or other instruments that have been collected, and systematically look at them and to say what are the changes, and then number 1, and then number 2, if there are changes, how does that actually, what's the meaning of those changes at the end of the day to the patient? So a lot of work still needs to be done in that landscape.
I was just going to add, thinking about Tiffany's comments and also just what you hear from parents, if a child doesn't get worse, that's also a really good thing if we prevent regression. And you think about the domains, right? It's very hard to measure cognition and language and these things. But then there are all these behaviors that are a little bit easier to measure and may even change in a shorter period of time. And I think we have a lot of opportunities to kind of refine existing measures that kind of capture psychiatric symptoms and could be validated in PMS.
Maybe picking up on that, I guess for different interventions, the outcome you're looking at might be different. Some could be specific to catatonia, let's say, which may have a very symptomatic therapy versus, change in the expression of SHANK3, which may have a different set of behaviors or changes you may expect. I was wondering if you could talk a little bit about sort of the risk benefits of the intervention itself, because a small molecule, that has relatively few side effects versus another more sort of invasive intervention might have different risk-benefit ratios. So I was wondering if we can talk a little bit as a group about sort of fit for purpose of these measurements and how we can design them for the purpose that they're aimed for. Who would like to take that on first?
So let's talk about sleep, right?
Mm-hmm.
I mean, that's an easy one because we have lots of sleep medicines. We don't know what works in Phelan-McDermid syndrome, but there, the risk is relatively low. The gain is extremely high. That's relatively low-hanging fruit.
Mm-hmm.
But then you talk about sort of cognition, language. There, to see improvements, I would think people would be willing to undergo greater risk. And obviously it comes with, significantly greater reward. Does that sort of answer your question?
It starts to, but
Yeah.
love to hear from the other.
I disappointed him, but.
Well, I think part of what you're getting at, Mustafa, is that it's so exciting that these therapies that we're talking about are what we call mechanism modifying. I mean, actually, hopefully helping genes be expressed that haven't been. But that's a whole new framework for the FDA that they're trying to tackle in many ways. And I'm only talking about the FDA as getting it out there, right? But regulatory and what that means. So, I mean, Latha's point about that we're tackling development instead of just getting rid of one little symptom is a big problem. But also when we truly, I mean, this is where it's so great that the basic people are here with the clinical people. But do we know what the mechanism modification actually is and where that's going to be? Is it going to affect just sleep? Is it going to affect all of development? At what rate? When's the best time? So all these things that we need to kind of guess at. And it's a chicken and egg issue, right? You've got to do the trials to figure it out. But I think that makes all of this really, really hard because, we can't have an endpoint that's development, right? We've got to get more specific. And I think in this case, it's the frontiers, right, for what it's going to be.
I can add to that too, building on there's comments. So if you're targeting sleep, we have great endpoints for sleep. We know how to measure sleep. But if you're targeting a if you have a disease-modifying therapy, we want to change PMS as a whole. It's across many symptoms, behaviors, skills, etc. Risk-benefit aside, how do we know it's working? How do you move the disease as a whole? What endpoint are we going to use? I don't have an answer. It's sort of a question. The way I often suggest is thinking about what Alex put up on his slide, where you have an indication-specific CGI, where you can have the symptoms of relevance to the disease, the symptoms of relevance to your mechanism of action, and you can, you get a summary score. It's not a mathematical composite, because FDA doesn't and I appreciate you're not gonna get a mathematical composite approved, but you can get a measure of disease severity with one score. So it's just something to think about.
I want to open it up to the whole group. With this background discussion, I would love to hear from others what their questions are, what their comments are. Yeah, go ahead. Back there.
No, I was
Oh.
going to say in adjacent therapeutic areas like in Rett syndrome, for example, one of the reasons that
Do you want to use the?
Sorry. One of the reasons, like in Rett syndrome, why I think clinical development was accelerated and we have an approved treatment is they did a really nice disciplined job with regard to creating an infrastructure around standardized ratings, for instance, standardized clinician ratings, and then also did, I think, a rigorous job at policing placebo responses. It was beginning to emerge and corrupt the integrity of the data, creating more untoward variability. So this is just sort of a point that it'd be great in this therapeutic area to begin to think about that as well in terms of methods for getting everyone on the same page, especially if clinical rating scales are where we're going to lean on, although I know EEG and the visual evoked potential are also going to emerge, but it's sort of the nemesis, it's been the nemesis in related disorders, this idea of intra-rater variability, placebo response, expectancy, gratitude, and so forth and so on. So it's just sort of a comment about moving forward, about trying to build a robust rating culture and here in this therapeutic area. And then the other comment I was going to make, and it's not just, sorry, it's not a question, but in other regulatory agencies, MHRA, in the United Kingdom, for instance, they're far less concerned about a substantial track record of validation. And I've had direct, repeated regulatory advice that says if this instrument works in the assay of this clinical trial, as long as there's other collective data elements that indicate it's not false discovery, we'll take this on face value that this is legit as an outcome measure for this disorder as you've applied it in this experiment, and then they've moved on. So just be aware that some of that neurotic approach that's used by COA and FDA is not replicated in other regulatory agencies to their benefit. So sorry, that was just sort of two quick comments.
Other comments? Maybe I can ask a, oh, is it that one?
So my question is maybe, obviously, parent side here. So we're asking, you're asking us to volunteer our children who necessarily. Oh.
You have to button to turn it on. There you go. To be green.
Sorry about that. You're asking us to volunteer our children to do things that they're not always necessarily able to consent on, and then at the top, when that ends, our children, in my experience, it takes so long to learn a skill, which is so much more longer than what is typical, that if they actually do learn it and then they lose it, what if they learn to understand a little bit more of the world and that gets taken back from them? What kind of like protections are there for the children to be able to not lose that? Obviously, I realize this happens for other diseases and things that are health related, but in this case, my child functions around 10 months old. Now he's able to function at 1.5 years. Like, that is a huge amount of growth and development to lose and watch them lose. What are the things that are in place for the mental well-being of not only the person, but also the family to have to give that back, to be like, trial's over, sorry.
Yeah, that's a great question. It goes to the ethics of designing these trials. Thank you so much for that question. Anyone wants to take that on?
So this is a question that I get asked a lot. I mean, if you get access to an experimental treatment in the context of a clinical trial and your child makes gains, what happens after the trial's over? Depending on what the treatment is, there are options. There's compassionate use programs. Sometimes these experimental therapeutics are commercially available and you have to figure out a way to get them, but sometimes they're not, and you don't have any guarantees. My experience has been that the gains made in the context of trials are sustained for the most part, but we don't have really good data to support that. It's just that the trajectory of gains kind of plateaus, right? So the benefit of the medicine doesn't continue on. And we're talking about small molecules now. We're not talking about, disease-modifying treatments. I got asked a related question that I'll share just last week about the gene therapy. What happens if my child gets so much better that they're aware at how disabled they are, right? Because that's something. If you're all of a sudden kind of turned on in a way that you're able to experience your deficits, that's horrible. I mean, that's really intense, I would imagine. But in some ways, it's a good problem to have, right? Because that means that they got better. So these are all the things that we're struggling with. I think the best that we can do is just be very, very mindful. And this is what the informed consent process is, right? We talk about all these things when we start clinical trials. And yes, the children themselves don't consent, but you as the parents do. But it's impossible to have guarantees. But it's a great question. Thank you for asking.
Go ahead.
Oh, could I just add one more thing
I'm
to
sorry.
that? I think also in clinical trial design, I think it's also important to have a check-in period after the intervention is over to check in with the families and to get a sense subjectively, were there qualitative improvements that have gone away or no longer persistent? That information can be useful as in future designs, but I think that should just be part of clinical trial design as well.
Thank you. So right now, I think, one of the critical gap is, we don't know which drug will work, and doing a behavior-based clinical trial will be very long and costly. And ideally, there will be some kind of really robust, readout that in small cohort you can have a proof of principle trial. When I talk to pharma, they just think, they've not a really lot of successful trial in ASD, and it's so variable and it's so heterogeneous. So if I have something I ask you right now, is there any biomarker info SHANK3 or any other ASD you are confident that it can be used as for a small cohort? If not, what it will be in the near future can we develop?
The easy question.
So confidence is a high bar, but that's exactly why we're working on these electrophysiological measures, because as long as we can show that they are connected to clinical outcomes and the readout is early, then that's evidence of at least engagement and the potential for change, even if it doesn't necessarily go downstream to behavior just yet. But that's exactly what we're working on. I mean, I couldn't agree with you more. Like we don't want to do 300-person trials for 2 years with only behavioral outcomes and discover that we failed. We want to be able to have some early predictor of treatment response. But yeah, EEG, and I'm as confident as I can be. It's a great story, but we have a lot more to do. And April's like, yeah.
And I think we're going to hear more presentations in the afternoon.
We have one question, but on the topic of biomarkers, we have a lot in block 2, which is all about translational, and then in block 3, I think it's when we have the EEG biomarkers. So we're going to talk about that a lot.
So one last question for this session, and I'm sure more questions will come up as we go on.
Thank you. Just following up on what the earlier comments about the regulatory frameworks in other countries. Are you guys, generally, are things worked on in kind of a US bubble, or do you guys look at, possible ways of speeding up the FDA approval process if you're able to approve this in the UK, in Canada, in Australia, in France, in those types of places?
I mean, we have some company people here that could answer that better. From an internal pilot, develop preliminary data and then have other people, really, move it along. It's an international effort, definitely. and feedback from Canada and feedback from Europe is extremely valuable, even if it's just being applied in the moment to the FDA. Is that accurate?
I think the developmental Synaptopathies Consortium is a US-based effort that the NIH is funding. But the data is available for us to share with anyone, and the consent forms are such that we can share the data with academic or industry partners anywhere. So there's no limitation for that first purpose. I think there are similar efforts in the Europe too to look at SHANK3 patients. I don't know what size they are and what stage they are currently, but I think in terms of data collection, it's been available globally.